governments, and universities has been developing potential vaccines at a pace and scale unparalleled in human history. It is a testament to the power of science to serve the common good in a time of shared crisis. And yet, according to Pew Research Center, between May and September of this year, the fraction of adults who voluntarily would agree to get a vaccine has plummeted from 72 to 51%. This is an alarming erosion of trust in the integrity of a process that we hope and believe will produce an effective and safe vaccine in record time. Our goal in convening this symposium with our partners at the University of Washington is to shine a light on every facet of vaccine development to ensure that this process is guided by the most stringent principles of sound science and only sound science. Because we know that even the best vaccine is only effective if people trust it and ultimately agree to receive it. We are fortunate in this endeavor to have the leadership of the Johns Hopkins Bloomberg School of Public Health, including Dr. Chris Beyer and his colleagues across the Bloomberg School, at the Center for Immunization Research, the International Vaccine Access Center, the Center for Health Security, and the Johns Hopkins Vaccine Initiative, scholars and scientists who are bringing their vast expertise to bear on this complex process. They, along with so many others joining us today, represent the very best of our great public and private research universities, institutions that have long borne the sacred responsibility of honing the scientific method, of advancing discovery across all fields of human knowledge, of safeguarding, protecting facts for the public good, and of creating opportunities for conversations such as this one, in which critique is embraced in order to refine and strengthen ideas. We know that science done right is important as science done quickly. These principles continue to define the indispensable role of universities in democratic society. The philosopher Hannah Arendt captured it so well in 1967 when she wrote that the chances for truth to prevail in public are greatly improved by the mere existence of universities. And I couldn't ask for a better university to partner with in this undertaking than one of the country's truly great public institutions, the University of Washington, and its quite extraordinary leader, President Anna Marie Kause. President Kause, over to you.
Thank you so much, President Daniels. I'm so honored to join you as well in this very, very important undertaking. The COVID-19 pandemic has presented us with challenges unlike anything in our lifetimes. The stakes simply could not be higher. All over the planet, lives and livelihoods hang in the balance. We urgently need working vaccines for COVID-19. Just as importantly, we need public trust in the scientific process that produces those vaccines. Fostering trust in science is essential for the pandemic we face today and for those we may face tomorrow, because COVID is not the last public health crisis that we will face. Trust in institutions and in each other is the bedrock of functioning democracies. And with today's proceedings, we seek to build and strengthen that sacred public trust. Research universities fill a unique and critical role in advancing knowledge and discovery for the betterment of humanity. We're dedicated to the pursuit of truth, and that requires devotion and strict adherence to the integrity of the scientific process. Universities are mission-driven. At the University of Washington, we define our mission as impact that serves the public good. This is a commitment and goal shared by our colleagues at Johns Hopkins and at universities and research institutes around the nation and world. The University of Washington and Johns Hopkins are both leaders in the fight against COVID-19. One from the public sector, the other from the private, one from the East Coast, one on the West. And today we come together to stand arm in arm with the extraordinary teams and individuals leading the work to produce effective COVID-19 vaccines. And we're calling for preserving the sanctity of the science behind their work and for the scientific process to proceed at the speed of integrity and accuracy, not politics. Today, we're going to explore the complex issues that will bring that integrity and accuracy to the outcomes of this process. How to structure better clinical trials, how to ensure equity and diversity in recruitment into those trials, and to provide proper regulatory oversight. How do we make sure that the distribution of the vaccine is guided by ethics and equity? How do we communicate clearly to a broad array of audiences? We're asking everyone, policymakers, scientists, pharmaceutical companies, the media, technology companies, and the public to unite around a common goal, the creation of working vaccines that have earned the trust of the public. And now it is such a pleasure to introduce someone who's earned the public's trust with his unflagging commitment to truth and public education. Dr. Anthony Fauci is the director of the National Institute of Allergy and Infectious Diseases at the National Institute of Health. He's become a household name as the voice of reason and authority in our national effort to stop the spread of COVID-19. As a scientist, he's devoted his long and distinguished career to learning, discovery, and truth. As a public servant, he serves not just our nation, but all of humanity. He's probably done as much as any other individual to maintain the public trust in science. And we are so honored to have him as our first keynote speaker. So please join me in welcoming Dr. Anthony Fauci.
I would like to thank the organizers for giving me the opportunity to present in a brief presentation in this session on the science and structure of the United States government COVID-19 vaccine trial program. As shown on this slide, I'm going to talk about the overall structure of the U.S. COVID vaccine program, but I'm going to focus a bit more on the NIH role, given my perspective as an NIH person. I'm going to divide this into 4 separate brief presentations. The first regarding the U.S. government's vaccine development stakeholders. Very briefly, who are they, or who are we, and what do we do? As shown here of the different stakeholders, the NIH, particularly NIAID, the institute that I direct, we do basic and clinical research on vaccine candidates. Our close colleague in this is the Department of Defense, who also does basic and clinical research on various vaccine candidates, as well as some limited manufacturing and advanced development specifically designed for their warfighters. ASPR, as well as BARDA, which is a part of ASPR, are involved in advanced clinical development and manufacturing support via a variety of contracts, and ASPR does oversee the Strategic National Stockpile. The FDA, the primary regulatory agency, advises on data requirements for each stage of vaccine development, as well as reviewing the preclinical and clinical data packages for potential authorization or licensure. And finally, the CDC, who via Advisory Committee on Immunization Practices recommends who is vaccinated, when, and with what vaccine, as well as shaping the prioritization for immunization when quantities are scarce. This is the pre-COVID-19 U.S. government vaccine development model, which we have actually employed and adapted for what we are currently doing today. As you can see, most of the early concept and product development resides with the NIH, who together with BARDA is involved in the advanced development, which is the major mandate of BARDA, together with the commercial manufacturing capability of industry. So BARDA involved with industry is very, very important, as well as then the final regulatory review of FDA. But please note on the slide that industry, as well as FDA, get involved in the process right from the very beginning as manifested by the NIH's role in collaborating literally within the first days of the COVID-19 outbreak with industry. And the FDA now, and the model that they employ today, is very much involved in consultation with us, with not only the NIH, but with BARDA and industry, right from the very beginning of the process. And this process indeed has worked very well. Then there's Operation Warp Speed, which is the overall coordinator of the effort, led by Moncef Slaoui, who you'll hear about shortly, as well as General Gustav Perna. I'll leave the discussion of Operation Warp Speed to Moncef Slaoui, who I believe will follow me shortly. Going to the second major group is the basic research. Let's talk about the novel platform technologies that have actually now been employed. This is particularly important because several of these do have this platform technology, such as the mRNA that we've seen in at least 2 of the vaccines, the viral vectors, the recombinant proteins, the adjuvants that are involved with the recombinant proteins, the virus-like particles and nanoparticles, which will likely be used in other vaccines. Then you go to immunogen design. This is something that really dates back to the work that has been done with HIV vaccine and immunogen design, structural biology, using a variety of modern techniques such as cryo-EM, has allowed us to very, very carefully and specifically delineate the conformational structure of the spike protein, which is very important in the development. This is a slide shown in MERS vaccine that was used, but we have now extrapolated that to the current COVID-19 vaccine. And then there's clinical research. We have networks in clinical research that we have actually built decades ago associated with our work on HIV/AIDS. This is a timeline from the very beginning of the HIV/AIDS outbreak from 1983, when we put together the treatment evaluation units, the original ACTEUs, which ultimately became the group called ACTG. Then there was the Prevention Trials Network and the Vaccine Trials Network. All of these now are playing a major role in our attempts to develop therapeutics and vaccines for COVID-19. Then there's the issue of, can you do clinical research in an outbreak setting? This is some of the history of how we've evolved to where we are today. When research was done with the H1N1 vaccine trials of the pandemic of 2009, the Ebola outbreak in West Africa, the Zika outbreak in South America, Ebola again in the Democratic Republic of the Congo, and then the SARS coronavirus 2, which is overseen now by Operation Warp Speed vaccine trials. So we have a clinical research apparatus that has performed scientifically sound and ethically sound research in the context of an ongoing outbreak. And in fact, the National Academy of Medicine did a post-Ebola study and wrote a report in April of 2017, which emphasized that the core principles of science and ethics in conducting clinical research does not change when that research is conducted in the context of an ongoing outbreak. And the randomized clinical trial are ethical and appropriate and are the most efficient and reliable way to determine safety and efficacy. Let's move on to community engagement, something that I have now had decades of experience in, as shown by this interesting historical slide. It goes back 32 years in the beginning of the outbreak, where the community felt they were not adequately represented in the discussions of the kinds of clinical trials that could be done. And this was an eye-opener for me. It was an article that appeared on the front page of the San Francisco Examiner in June of 1988 by my now, years later, who became a dear friend who unfortunately has recently passed, namely Larry Kramer. As you can see, the title is, "I Call You Murderers, an Open Letter to an Incompetent Idiot," Dr. Anthony Fauci of the National Institute of Allergy and Infectious Diseases. That was 32 years ago. He wanted to catch our attention. He and the activists, namely ACT UP, felt that we were not doing enough. We were not paying attention to their needs. We did not engage them well. Here's an example of a demonstration, as you could see from the signs And remember, Ronald Reagan was president at the time. Again, they felt we were not engaging them properly. This is a Academy Award nominated film, How to Survive a Plague, which was talking about the evolution of the then to become successful community engagement. The person on the slide is a young man, then now my colleague and very dear friend, Peter Staley, who has emerged from an adversary to now one of my close personal friends. Well, I'm telling you this story because what this led to was our developing community programs for clinical research on AIDS, as well as community advisory boards on virtually everything we did. We firmly engaged the community. That is trying to be done now with the SEAL program, the Community Engagement Alliance Team, which is a pan-NIH entity which leverages our long-standing clinical trial network engagement that I have mentioned to you just a moment ago, as well as the COVE-PN Community Engagement Working Group, which is working on that well-established community engagement program. And finally, the COVID-19 clinical trial approach. This is a list very familiar to you now of the multiple platforms, the multiple companies, and the various stages of clinical trial, with 5 of these now engaged in either phase 2 or mostly phase 3 trials. We have put together what I call a strategic approach to COVID-19 vaccine development with harmonization of the multiple trials with common data and safety monitoring board, common to the extent possible primary and secondary endpoints, as well as common immunological parameters that are used for correlates of immunity. This is a map picture of the location of the NIAID COVID-19 prevention network, which is now being actively used in the prevention trials that we are now implementing. And finally, I'll close with this last slide, which if anyone has any interest in looking after and looking out to see what the prevention capabilities are, what the trials are, and importantly, if you actually want to volunteer or at least be considered as a volunteer for one of these prevention trials, particularly vaccine. So I'll close there and thank you very much for giving me this opportunity. Thank you.
To introduce our next speaker, Dr. Mansuf Slaoui, who is currently serving as Chief Advisor for the United States government's coronavirus vaccine development program, Operation Warp Speed. Dr. Slaoui has devoted his life to vaccines, which, as he is often noted, stand among the most effective public health interventions in existence. From his days as a professor at the University of Mons in Belgium to his position as Chairman of Research and Development and Global Vaccines at GlaxoSmithKline, where he oversaw the work of thousands of research scientists, to navigating in this moment one of the most difficult and scrutinized jobs in the world in search of a COVID vaccine, Dr. Slaoui has been unequivocal in his desire to save lives through vaccination, but always steered by scientific fact. We thank Dr. Slaoui for taking the time to join us today. Over to you.
Thank you very much, Mr. President Daniels. Ladies and gentlemen, very, very nice to talk to you today. Thank you for the invitation. I'm going to try to be very brief and describe to you what Operation Warp Speed is, how we're organized, and how we go about supporting development of vaccines and therapeutics. I'm going to focus on vaccines today. I'll try to do this in 10 or 15 minutes maximum. The operation is a team with a mission to integrate the great work from various agencies in human and health services, such as the NIH, NIAID, such as the CDC, such as BARDA, ASPR, and also agencies from the Department of Defense, in particular DARPA, or the logistics organization and a contracting organization, and really the might of the US Army in terms of infrastructure and logistics. Integrating these capabilities and capacities that are within the US government with those of the private sector, particularly from those companies, biotech or large pharmaceutical or medium pharmaceutical companies, that the programs of which we have selected either vaccines or therapeutics, and also with the work of academic centers, and in particular, the COVE-PN network that are a critical part in the execution and running and design of the clinical trials for the vaccines in question. There is a very small leadership team, and we have dedicated project team for each one of the programs that we have in the portfolio. Our role is to select programs, and once we've selected them, to enable them, finance them, advise them, facilitate, accelerate them, because our mission is to deliver approved vaccines to the American people before the end of the year and in enough quantities so as to immunize the US population potentially by the middle of 2021 or slightly later. We elected, in order to manage the risk and at the same time provide us with opportunities to have different vaccines with different product profiles for different subpopulation targets, we elected to build a portfolio of programs based on the selection of 4 platform technologies for vaccines. And we selected these platforms on the basis, on our judgment, for the best balance between speed of development, likelihood of efficacy, expected safety profile, scale up and industrial manufacturability, and also capacity of their private owners, the companies that we sponsor, to execute, because the operation is not the executor, it's really the enabler for the executors. We've selected, as I said, 4 platform technologies, and we elected to have 2 vaccine candidates within each platform technologies, again, to hedge our bets. Of these, 6 are public. 2 are based on messenger RNA, the vaccine from Moderna and a vaccine from Pfizer. These 2 vaccines are now in phase 3 trials. They have almost completed their recruitment, 30,000 subjects for Moderna and about 45,000 for Pfizer. And we expect them to read out or have a first look at their efficacy outcome within the next 7 weeks. Nobody can really say when, but the expectation would be that this would happen between the month of November and December. The second platform technology we selected are non-replicating live vectored vaccine. These are either chimpanzee adenovirus used by the AstraZeneca and Oxford University vaccine, or human adenovirus 26 used by Janssen's J&J. These 2 vaccines are also in phase 3 trials, less advanced in terms of recruitment than the first 2 vaccines. And one of them, the AstraZeneca vaccine, is on hold at the FDA. It was also put on hold in other countries where it's running phase 3 trials, the UK, Brazil, South Africa, Japan. But those holds have been lifted. The lift has not yet taken place in the US. Because their recruitment is at an earlier stage, the expected earliest readout for efficacy of these vaccine trials is somewhere in the beginning of 2021, probably January. And then the third platform technology are adjuvanted protein, recombinant protein vaccines, a vaccine from Novavax and a vaccine from Sanofi and GSK in partnership. These vaccines are in phase 1 and phase 2 trials, and they are expected to start their phase 3 trials probably by late November. And again, because of that later start, they will expect to have their phase 3 readout on efficacy somewhere towards the end of the first quarter of 2021, March or something like that. Supporting these, and then a fourth platform that we haven't named what vaccines are in it, but it's a platform for live replicating vectored vaccines that have the potential to be either a 1-dose vaccine or an oral vaccine. Those are not yet effectively in the clinic and are unlikely to achieve clinical efficacy prior to the middle of 2021. To support these programs into preclinical and then early clinical and then phase 3 trials has been a monumental effort by all players, the companies, operation, the various agencies, the academics, and the CoVPN network. And I should say we're reasonably pleased with the progress. It's been really fast, and we expect to have 3 waves of efficacy readouts over the next several months. First wave with the RNA vaccines, imminently, November, December. A second wave with the non-replicating vectors vaccine in January or February, and then a third wave with the protein adjuvanted in March, April. That's great and excellent, and everybody actually expect then that filing for an emergency use authorization or BLA would take place. But that, of course, is only half of the story. The other half, equally important and frankly even more complicated to deliver, is to scale up and to industrially manufacture these vaccines. Producing a few hundred doses, a few thousands or tens of thousands of doses per batch of vaccine is one thing. Producing millions or tens or hundreds of millions of doses of vaccine, where you make sure that the first dose and the 100 or 500 million dose of vaccine is the same, consistently the same, is a very different story. We have invested enormous efforts and finance and technology in supporting the companies directly and in fact much more closely to scale up and manufacture the vaccine doses and do it in a way and at a pace that allows the vaccine manufacturing speed to match that of the clinical development, because it's obviously useless to have lots of vaccine doses and no clinical efficacy, and even more dramatic to announce great success for clinical efficacy and have no vaccine doses to immunize and respond to the expectations of the population. So an update on that for the messenger RNA vaccine, Pfizer and Moderna. This is a completely novel technology, very little experience with it and with scaling it up. However, fortunately, it's a semi-chemical process, much less complex than the biological processes. I don't want to downplay it, super complicated, but less complicated and more predictable. I'm very pleased to say that thanks to the enormous focus and efforts that both companies have put behind this and the support that we provided them in every possible dimension, from providing government dictate, I would say, on supply of raw materials and equipments, to hiring of people, to training, to etc. They have now both been able to achieve industrial scale manufacturing of the vaccine. We are in the process of stockpiling vaccine doses in the single-digit million doses in the months of October and November, and then in the tens of millions of doses in November, and in the month of January onwards, we have between 20 and 30 million doses of each one of these vaccines manufactured at scale. The second family of vaccines, as I said, are the non-replicated vector vaccine from AstraZeneca and J&J. The AstraZeneca vaccine process has actually now been scaled up at industrial scale, about 2,000 liters. The challenge we have, frankly, is that while the clinical trials in the US are early, the phase 3 trial is in hold, as I said earlier, there are phase 3 trials ongoing in the UK, Brazil, and South Africa, and those trials may deliver their readout or first readout on efficacy somewhere in late October or November. And at that time, we will have very few doses of vaccine to be able to, if a decision was to approve that particular vaccine, we'll have very few doses to immunize. We're working very hard to accelerate the manufacturing and stockpiling of this vaccine. We will start to have a few tens of millions of doses as of January onwards. And there have been significant changes in the scale-up process to the way the vaccine is manufactured. So we may have to do some clinical bridging between the early vaccine doses that were used in the early clinical studies and the stockpiled vaccine. The J&J vaccine process manufacturing is progressing pretty well. It's about 6 or 8 weeks behind that of AstraZeneca. Both of these vaccines are being manufactured at a contract manufacturing company called Emergent and based in Baltimore, Maryland. We have worked very hard to equip that company, to source it with talent, train them, raw material, the whole gamish. Good progress, but very hard work. The protein vaccine are the most complex to scale up. We have to produce the protein and have an integral three-dimensional structure. One of the proteins have its transmembrane region and therefore is much more difficult to separate from any other proteins in the milieu. However, because their time frame for clinical efficacy is somewhere in the first quarter, for these vaccine, we will actually have vaccine doses available prior to the expected availability of efficacy trial will start to stockpile these vaccines in January. They're being produced in manufacturing facilities in North Carolina and Texas for Novavax, and in Massachusetts and New Jersey for the Sanofi vaccine. All in all, we have actually created a network of 25 manufacturing sites across the US, all of them in the US. About half are involved in what's called the drug substance manufacturing or the bulk manufacturing, so the fermentation, making the protein or the virus or the RNA. And the other half is involved in what's called the drug product manufacturing, which is the sterile fill, finish in vials of the vaccines that needs to have, and imagine 300 million doses is not something that you can vial very quickly in a sterile environment. This network is working very, very well, and we're very, very happy with it. The one learning message that we came to as part of this process was to recommend to the companies that we are supporting that if they achieve efficacy demonstration, while there are no vaccine doses available at industrial scale at several million doses to be able to immunize at least a relevant fraction of the population, that they should refrain or at least consider refraining from filing for an EUA, because a population would have major disappointment on the expectation for availability of the vaccine. Finally, just a few words to say the operation is also involved with the distribution of the vaccines and in a way in the allocation of the vaccine doses. We very early worked with the NIH and Dr. Francis Collins to identify the best possible party that can help us with a science-based, ethic-based, data-based assessment of how best to allocate vaccines in the setting of an epidemic. The National Academy of Medicine was identified and did a tremendous, great job that was just published last week. And I was very interested and pleased to see, for instance, that in their 3 phases of recommended immunization in stepwise way, the first 2 populations represent about 80 to 90 million subjects in the USA that can be subdivided in 2 subgroups, one of 30-some million and one of 50-some million subjects. The reason I say that is we feel comfortable that within the next month, month and a half, 2 months, we will have 1 or 2 vaccines, the RNA vaccines, that will read efficacy and for which we will have enough vaccine doses as of November, December to immunize with 2 doses, 30 million subjects first, and then in December, January, immunize 50 million individuals and start to significantly impact this pandemic into the country. And then things will continue afterwards. I'm amazed actually to just take a step back and realize that the sequence of this virus was described on January 23rd, 2020, which is less than 10 months ago. And we're sitting here talking about programs in phase 3 trials in industrial manufacturing. And probably by the time, well before the time 1 year has gone by, we are likely to have effective, safe, and largely available in doses vaccine. I think that really speaks to the enormous commitment that all the players have had behind this crisis on the one hand, but also to the great advance of science and technologies that underpin vaccine design and development, whether in academic setting or industry. Thank you.
Well, thank you so much, Dr. Slaoui. This is so incredibly exciting and promising. The effort that you're leading in Operation Warp Speed is of critical importance, not only to the people of our nation, but as you have so aptly said, to the whole world. We join you in believing that science's highest calling is to advance the public good. And as Dr. Slaoui has noted, it is paramount for the safety of millions that the vaccine trial process be allowed to proceed with all the appropriate procedures, reviews, and oversights, even within the context of an accelerated process. Those checks aren't just critical to the development of a safe and effective vaccine, although they are. They're also essential to maintaining and inspiring the public trust. And I so much applaud you, Dr. Slaoui, for your unequivocal pledge to resign rather than to tolerate political interference in this development process. That is so incredibly admirable. Thank you. The absolute necessity of a vaccine process endowed with scientific integrity is at the very heart of our mission. And it's why President Daniels and I have joined together to host this very important series of conversations. It's such a pleasure to be working with you, President Daniels. Back to you.
Thank you, Dr. Kase, and all our colleagues at the University of Washington for your partnership and leadership in convening this important conversation. I'm grateful to our speakers for their time and for sharing their insight into our collective efforts to find a vaccine at this critical juncture in human history. And it truly is historic. Yet, as Dr. Fauci said, our current search for a vaccine is and must be built on a firm foundation of efforts that have established efficient, effective, and ethically sound approaches to such development. In a pandemic such as this one, we cannot abandon such approaches, but must double and redouble our efforts to adhere to them ever more sedulously. We only have to look at global efforts to combat Zika and Ebola to see that the evidence supports the system's past successes. And we can trust that evidence, when it is validated and then delivered by someone like Dr. Fauci, who has quite literally been in the room where these developments have happened for nearly 4 decades. It's an extraordinary career. We are delighted to have the chance to hear this afternoon from leaders of our present effort. Thank you again to all our speakers and Dr. Kase. Now, I'd like to turn things over to Dr. Chris Beyer, Desmond M. Tutu, professor in the Department of Epidemiology at the Johns Hopkins Bloomberg School of Public Health, and a senior scientific liaison to the COVID-19 Vaccine Prevention Network, and I might say, a close friend. Chris, over to you.
Well, thank you so much, President Daniels, and ladies and gentlemen, a warm welcome to everybody, to all the attendees of today's symposium on protecting the integrity of the COVID vaccine development effort. As you've heard from Dr. Slaoui, this is truly an unprecedented undertaking. As Dr. Daniels kindly noted, I'm serving as your moderator for the 4 panel sessions that are to come. Each panel, we've tried to bring together really authoritative voices to discuss and debate the critical aspects of vaccine trial outcomes and vaccine trial integrity. The first is going to focus on the conduct of the efficacy trials themselves and the critical importance both of having multiple trials and of diversity and inclusion in those trials. The second will address the issue of regulatory integrity, with a particular focus on the FDA and the issue of potential emergency use authorizations that Monsef spoke to for the COVID candidate vaccines. And many of you will know that this is in the news yesterday and today, with the FDA reasserting that it is going to maintain the highest standards for these EUAs. The third will address integrity in communications and feature science reporters who are covering the trials and the pandemic. And the last will address the most important issues likely facing us if and when we do have safe and effective COVID vaccines, the issues of allocation, access, and equity. After these sessions, Dr. Ellen McKenzie, the Dean of our School of Public Health, and Francis Collins, the Director of the NIH, will close out our programming. So let me then introduce the first panel of the afternoon, Protecting Scientific Integrity in the Design and Conduct of the Trials. This session is going to be co-chaired by Ruth Karen, Director of the Center for Immunization Research at Hopkins, and Dr. Keith Jerome, who's professor and head of the Virology Division at the University of Washington Medical School. So keeping our theme of Hopkins and Washington. We'll begin with a keynote by Professor Larry Corey, who is the co-chair of the COVID Vaccine Prevention Network, the CoVPN, and professor of medicine and laboratory medicine at UW. And then Larry will be joined on the panel. He'll join the panel, and the panel will include Dr. Michelle Andrazik, who is leading community engagement for the CoVPN, and they will be joined by Dr. Joseph Millam, who is a bioethicist at the Clinical Center, the Fogarty Center of the NIH. So Ruth and Keith, over to you.
Thank you.